Hey everyone, this is Brandon Monnier, and this is a supplemental video that is going to go over the workshop that was hosted last year at the ILSI annual meeting in Senegal. This was around the beginning of October. And so this is going to be essentially a two-part video because this was 2 days of teaching. And basically this is just like going to be a recording over what we covered at the workshop for, your own personal streaming and personal reference and, studying. So the first day we went over several key objectives. One of it was kind of like a brief overview of JupyterLab and the JupyterHub. So where I am right now, this isn't, so at the workshop, we were on a workshop server. This material can be found on the main ILSI hub, which I believe a lot of you already have accounts for. If you don't have accounts already, please talk to either me or Bethany Econopoli or Francisco Augusto Perez. And so what this is essentially, this is JupyterLab. This is a place to host a variety of different notebooks, but it also gives you, more of like a greater interface to file structure and things like that. So this is kind of like, we have different areas of things that are able to be shared across the hub in terms of data, in terms of notebooks and scripts and other things relating to that. So the materials that are found here today are in the shared folder. This is under Commons. And so when you go into Commons, you have these 3 different folders. We have data, notebooks, and source. First thing we're going to do is we're going to go to notebooks. And we notice we have a lot of different subdirectories in here. Where these are found are in this interoperability workshop rTASSEL. I'll make this a little bit bigger for now. So this is the interoperability workshop rTASSEL 2022 folder. If we double click that, you'll be provided with the Jupyter notebook that I'm going to be going over today, and then also part 2, so that'd be day 2 of the seminars for the notebooks. And then also we have a PDF and some additional materials pertaining to Fieldbook, because Trevor gave a nice little overview of the Fieldbook component, and then some introductory materials in a PDF format, which you can view either also within the JupyterHub, but you can download this and view it on your local computer if you want to. And this kind of gives you an overview of what we've done that day. If we go back up a couple of levels back to the Commons directory, we also have this data directory, and this is where the example data that we use for today, or for day 1 and day 2's workshop videos, will be found. So again, we have a lot of different shared subdirectories. One of the subdirectories is this workshop Senegal, and this is where that information is found. And so we'll have just all of the example data that we use here. You shouldn't have to really worry about going directly into each one of these different data directories, and at least in terms of getting the data, because as we scroll down through this notebook, all of this material will be in hard-coded paths for you to follow. So yeah. And so again, this is a notebook. This is a place that you can have things like text as found here in the form of Markdown, but you can also have blocks of code. In this case, we're running R. So just like running R locally, you can run R in these cell blocks, and then you can evaluate it and get results within the notebook. And so to do that, you would essentially, there's 3 different flavors. There's code, there's markdown, and there's also a raw form. But we've already had another workshop covering that, so I'm not going to really go into a lot of details. And while I go through this notebook, I'll kind of guide you through some of the different functions that may be of use to you. So another thing that we went over was rTASSEL and rTASSEL. So I'll give a brief overview of what that is, and then we're going to go through some analyses with rTASSEL. And this will be basically what we cover for this first video. All right, so moving down to the first cell block here. So you'll have this libraries needed. So these are R packages that we are going to be using for this session. A lot of these you've probably already heard of, like dplyr, ggplot2. This is for data manipulation. This is for visualization. We have like an extension to ggplot2, which is ggtree. That's going to provide us like enhanced phylogenetic trees. We have this magrittr package for pipes, which I'll show a bit later. Tibbles for tidy data frames in R. And then of course rTASSEL, which is what is going to be the main focus for this section. So I've already ran that. And basically if you ever want to run a cell block, you would make sure that you're already highlighted or click onto the cell block, and then you go up here, there's going to be this play button, so this triangle shape button right here. If you just click that and run it, or on your keyboard you just go Control Enter, or at least on my keyboard it's Control Enter, that will evaluate the code that is found within this respective code block. So I've already ran that, and you notice that each one of these packages has their own kind of like startup message. So we're attaching package magrittr, and then we're also loading rTASSEL, which was one of the last things that we loaded. Another thing that we're going to be using today is this helper function that might eventually be brought into another R package, and this is going to help us a little bit setting visualization dimensions of some of the plot output that we generate today. And so this is just calling, we're making a local function, so we're using the function method within R, and we're passing a couple of parameters, and this is going to allow us to set the width and height of our visualizations within the notebook, and that's going to be passed through this fig variable. But we'll go back to we'll come back to this later with further detail. So the next section that we're going to talk about is rTASSEL and what that is. So rTASSEL is a extension to TASSEL 5, which I believe a lot of people have already heard of. This is an extension to TASSEL that allows you to write things in R, so R-related methods and language. And what that's going to do is it's going to be interpreted using the TASSEL 5 backend. So anything that you pass in R is going to go into this TASSEL library to be evaluated, and then it's going to come back to the R environment. So this basically helps you, write things in more of like an R-based domain, and you get the performant ability and also access to all of the different methods that are found within TASSEL 5 without ever having to leave like a Jupyter notebook. And then this also allows you to interconnect this with other different packages, which we'll show later on in this video and then part 2's video. So TASSEL, again, is for a lot of different genetics analyses. So this is kind of like a broad general package that contains a lot of different components to efficiently read in genotype data, also phenotype data, being able to do association analyses, look at population structure, develop kinship matrices, and provide components that will be useful for things like genomic prediction and other types of linear modeling. So the first thing that we're going to do is we're going to set up some paths to some genotype data. The first thing that we're going to be loading in is a VCF file. So this is one format of genotype data. So this is a variant call format. And then the next type of genotype data that we're going to be loading in is this HapMap format. So Tassel can load in a variety of these, and you don't have to like specify, a specific extension. It automatically guesses based off of the data structure. So what we're doing here is these are the locations to the example data that we're going to be loading in. So again, I'm going to run that. So we can either click the run. So I'll click on that, and then you notice that this gets evaluated. So we get these two objects, this genopath VCF and genopath HMP, and then we're just printing those out to the notebook, and we're just printing out those hard paths, so those full paths to the actual text files, or in this case the VCF and HapMap files. So the next thing that we're going to do is we're going to take those objects, and then we're going to read those in to Tassel to make some native R objects. And so to read this data into rTASSEL, we use the family of read functions. And so we're going to be using this read genotype table from path. So we're going to be creating a genotype table, but we're going to be reading this data from a file path. And so that takes one argument, which is this path argument. And so you dictate one of these different variables here, and then that will be piped into this, or passed into this particular Tassel genotype, or Geno, HMP, and VCF objects. So if we run that, I'm just going to use Control-Enter on my keyboard now. And now what we can do is we can take a look at one of those objects. So what gets passed in is we get this genotype table, and then we create this Tassel genotype phenotype data set. And so it's going to have the class, or the name, the proper name of the object that we created. It's going to have the number of samples. In our case, this is 281 samples. We're currently calling this taxa in rTASSEL, but we will probably change that in the future, given the amount of confusion that there was in terms of taxa. So think of, and I'll just make a note right here, think of taxa is going to be equal to samples, sample IDs, essentially. So like the number of rows in this case. And then we have the number of positions. So these would be like the variant markers that are found in the genotype data. So in this example data set, we have 3,093. And then we have this third row down here, or technically this fourth row in this object, which is the how many different elements, so the taxa times the position. So it's a little bit under 870,000 different taxa, or different sample IDs by position markers. And then you have some information down here which indicates what has been loaded into this data set. So in this case, we've just read in a genotype table. So the genotype table parameter gets populated, but we notice we don't have any phenotype information. We can read in phenotype data similar to how we read in genotype data. So we can either read that from a path. And so we're just going to call this. And so this is the path to that object. But one thing I want to take a look at first is what that data actually looks like. So this is demo data phenotype tassel.txt. So I'm just going to double click on that. And the nice thing about the Jupyter Lab is that it's able to not only open up notebooks, but it's able to open up, just raw text files. So again, you can do a lot of things besides just run notebooks on the LC Hub. And so if I take a look at that, and I disregard these first two rows right here, you notice that the format of this type of file, so this is a Tassel 5 type phenotype / trait format file, and that you have essentially a delimited text file, right? So it's been delimited by tabs, and so that's what that little character is showing right here. This is just indicating a tab instead of just like a white space. So we're delimiting each one of these columns by this tab character. And so we have taxa, so again, sample ID. In this case, we have a little bit under 300 sample IDs. We have this location variable. So this would be like, let's say you plant a trial in location A, and then we plant another, selection of the same sample IDs in location B. So this would be like a categorical variable in your experimental design. And then we have 3 different measured traits. So in this case, we have ear height. So this is maize data. This is days to pollination, and then ear diameter. And then you have the different types of data that are found. So taxa would be like a name. Location will be, location A, location B, or whatever you want to distinguish in terms of your actual location. And then you have like the measured values. So these would be things that you would physically measure either out in the field or in a laboratory setting. And so going back to these first 2 rows, since this is TASSEL 5 formatted data, we need to specify this phenotype tag. So we have phenotype, and then we have these 2 angle brackets that are surrounding that phenotype indicator. So that's gonna denote that, oh, this is phenotype, this is trait data. We're gonna read that in as such. And then what we have are different types of data. So remember, in R, we can have different types of data in terms of like, a number. So going back to the workshop slide here, I'm just going to make a new cell. And to do that, what you can do is you can just press the letter A or B on your keyboard to either introduce or bring in a new cell above, so clicking A, or below, clicking B on your keyboard. And if you want to delete that, it's just DD. You press the letter D twice. Or you can, actually delete the cell by like right-clicking on the cell with your mouse or touchpad, and then you hit the delete cell button right there. And then you can also insert and insert cells above and insert cells below using the same methodology. So for this example, in R, we can have things like numbers. So we can have like the number 1, right? And that gets evaluated to number 1. We can have things like characters, like hello, that's a string. And then we can also have things like factors. So we can go like factor, and then we can go A again, B, B. And then I evaluate that. And that's because that needs to be passed as a vector. And so, yeah, we just pass like a collection of elements using this C component. And so what it will do is we'll print this out, but you notice that there will be this levels option because this is, 2 categories technically. All right, so we can have all of these different types of data. The same thing applies to, and I'll just kind of keep those there for now, but the same thing applies to TASSEL 5 data. So the different types of data that you can have are taxa. So this would be your sample ID. So this would be like, what are each one of your samples or observations called? And then you have location. So it's just like R, where you can have a factor. The same thing applies here, where you have different categorical variables representing different types of data. And then you have the kind of like base data type. And so this would be things like your actual measurements, the things that you're actually recording out in the field or a laboratory setting, something that is generating like a continuous variable. And then you can also have something else that's not shown here, which is covariate data. So this would be things that, if you're building a model, these would be things that would help, control for variation within the model. So things like, population structure in terms of principal components. If you're doing like an RNA-seq study, these would be things like peer factors, things like that. And those would be labeled as, you know, a covariate. But again, this example doesn't have that. We're just dealing with factor at the moment. So that's what that file kind of looks like. And so we can, you know, populate this new object called phenopath, and then we can read that phenotype from path, again, using another rTASSEL read method. In this case, we're reading that from path. We call this taskPheno object. And now you notice that we get a similar data object, but slightly different in terms of its representation. We have the number of taxa, in this case, the number of sample IDs. And now we have this phenotype table object that's populated. And we can see we have a little X to show that this component in this object has been populated with this phenotype table. And then we have an additional message below that genotype and phenotype table that indicates what are the traits that are found, or basically a better way to describe this is like what are the different columns that are being identified within this data set. So that is a phenotype, but we can also, what we can also do is we can take that same data, right? And this may seem like kind of like a lot of work to format your data like this. But again, if you're using TASSEL 5 proper, this would be how you'd have to format it. But the nice thing about like rTASSEL is that, you know, like R in and of itself, in addition to like things like factors and characters and numbers, we also have more complex data objects. So things like data frames. And so if you look at these two things, they're pretty much similar. And so the nice thing about R and rTASSEL is that we can actually take data frames that are, you know, read in R in the like your actual environment, and then pass those in to, you know, TASSEL. And so what I have right here is some additional data that I'm reading in. So this is like another data set, which is basically that same data set, but it's delimited by commas, and we've removed the phenotype tag and then this data type row. So these rows 1s and 2s have been removed. And then we're just reading that in using read.csv, and we're taking a look at the first 6 rows. So you notice that we have taxa, we have location, ear height, days to pollination, and ear diameter again. But now what we can do is we can read that same type of data set and using an additional member of the read family, which is read phenotype from data frame. So instead of like a path, so a location to a file that's found on your computer, you're reading this from a native R object, in this case a data frame. So the arguments that you pass through this is the phenotype df, which is what is the data frame that we need to load in. So in this case it would be phenodf. And then you need to specify a couple of additional arguments. So what is the sample ID column? In this case, the taxa column. We're just going to call that as a string. So this would be this column that's called taxa. Right? So we specify that column name here. And then we have the attribute types. And so again, going back to this data set, this would imply this column right here, right? So you just pass that as a vector. So besides the taxa column, we have the location column here. So that would be factor, because again, this is a categorical data set or data type. And then we have data. And so you can either write data out, you know, 3 times, or you can use the rep function within base R that will replicate that data word 3 times within your vector. And then you read that into this taskPheno df and then inspect it, you get the same thing. So we've read in the same type of data, but instead of, you know, from an actual flat file that has text, we're reading this in as a data frame in R. So for certain things, You may just need genotype data to analyze, or you may need just phenotype data to analyze. But if you're doing something more complex, let's say like an association study where you're trying to link a phenotypic trait to marker data and or covariate data, depending on what you're implementing in terms of your model, you would need to basically union or join this phenotype and genotype data together. And so we have yet another method within the read family that allows you to read both genotype and phenotype data at the same time, in case you don't want to go through reading in phenotype data separately and then genotype data separately. You can read it all together in as one object. So this is kind of like, you can take a path, it can take a data frame, or it can take like another rTASSEL object. So in this case, I'm just passing in that TASS pheno data set. And then same thing for the genotype data. So I'll just put a little comment here. This is phenotype info, and this is genotype info. This will be either the path or the object to the genotype information. So that would be that TASS geno. It's up here. Yep, this TASS geno HMP in cell block 5. So I'm just going to read that in. And so what that's going to do is it's going to combine both of the phenotype and genotype data sets together. So what it's going to do is essentially a intersect join. So it's going to find what's common in terms of samples that are found in the phenotype data and the genotype data. So you notice that the phenotype data has 284 sample IDs. The genotype data has 281 IDs. And so the intersection or the commonality between those two is 265 samples that have both trait information and marker variant information. So we call that up. So another thing to notice is that we have both the genotype table and phenotype populated in this data set, and we also have the different traits that are identified. So in this case, those different columns within the data set. And then this right here was just kind of looking at the object structure. And so if you take a look at this actual object, this isn't necessarily like, a full-blown native R object. What this is essentially doing is it's kind of like a collection, or you can think of it as almost like a list in R that's pointing to different areas within Java that have these TASSEL coded objects. And so for that, we have like our own custom getter and setter methods that we will talk about later to pull this data natively into R. And that's the next step right here, is like, how do we get this kind of like abstract representation of like the TASSEL library into R that we can then, pass to different pieces of software, different analytical methods that don't necessarily require rTASSEL specifically, but maybe something as like a more general object. So we have a lot of different ways of getting data out of these objects. So for these sections, I'm just going to be using this TASS geno pheno object. So the first thing that we're going to do is, we've read in either VCF or HapMap data into rTASSEL, and now what we're going to do is we're going to convert that into a R matrix. And so to do that, we can just use the base R method as.matrix to take that object and coerce it into a native R matrix object. So I'm just going to pass that into this genomat object, so genotype matrix for short. And I'm going to take a look at the first 5 rows and the first 5 columns within that matrix. So you notice that it's in this case, it's a 5 by 5 matrix because I'm just taking the first 5 rows and 5 columns, and it's of type int. So this is basically a number in R of 2s, 1s, and 0s. So the copies of the the alternate allele. How many copies of the alternate allele do you have within your data set? And so you notice that for the rows, you have each one of the sample IDs, or the taxa IDs. And then for each one of the columns, you have the name of the marker that was found in the genotype data. So in the case of this maize genotype data, we have this PZB, PZA type nomenclature, and that's just basically an ID for each one of those markers as you move across the genome. So now that you have a matrix, you can then, you could pass this to another piece of software, or you can use base R methods to do that. So that's one option that you can also do with rTASSEL. We can also coerce, or we can pull out the actual data frame from this phenotype data using this get phenotype data frame from the rTASSEL object. So this is a built-in method in rTASSEL called get phenotype df, or data frame for short. We can call that into this phenodf function, and we're just going to take a look at that. And so this is basically, we've converted it into this rTASSEL form and then reconverted it or exported it out as a data frame. So this would be, one way, like let's say you're dealing with data that looks like this TASSEL5 formatted data. This is a pretty simple way to convert that into like an R data frame. Then we can also get different types of summary information from each one of these objects. The first thing that we can do is, and this is where McGritter comes into place very well. So you can think of, again, this McGritter pipe, so this %, right angle bracket, %, component as kind of like an arrow, a directional arrow of how you're processing this data. Or you can think of it as like, flowing water or ingredients that are being passed into a function to make one component, and then that newly made component is then passed into another function to create, an even different component. So you can think of it as like flowing water, or like a factory or an assembly line that's making like, I don't know, a car or something, that you start with like the chassis, and then you add like the engine block, and then you add the wheels, and then maybe you add the paint, etc., etc. That's, these are kind of different ways that I like to think about, this McGritter pipe. And what's really nice about that is, we don't really even need to do this, but if we look at something like, this example right here, if we were to write that in base R, what you'd essentially have to do is you'd have to like invert this, and then you'd have all of these different, you'd be calling a function inside of another function, and then depending on how complicated your workflow is, you could have several different types of functions nested together to work on this initial object. And so to me personally, I think looking at something like this is a lot easier to digest than trying to interpret something like this, where you have to like work your way inside out. But just to kind of like prove to you that this, these two functions actually work together. So first example is, what exactly is this doing? So as the name implies, we're getting a summary table, but of what? In this case, we're getting the summary statistics of each one of the variant markers, or in this case, each one of the sites that's found within the genotype component of this task-pheno object. So I'm just going to call that, and I'm going to call the head because I don't want to call all 3,000 markers to my notebook. And so what you get is essentially just simple metadata on each one of the different variant markers. So you have the name of the site, you have its indexed number value within TASSEL, where is it located on the genome in terms of its chromosome, in terms of its physical position, how many taxa have that, what is the major allele, numbers of major alleles, numbers of, if there's more than just one or two, what are the other representatives in this case? And then we have like the proportion, like the number of gametes that are missing that particular marker, and we can calculate proportions. How many of those samples that have that marker are heterozygous? What's the proportion of that? And some additional things that you can populate later, but we're not going to really talk about. So that is the site summary table. So in case you want to like look up each marker, how much is it being represented by each one of these different statistics in terms of like what's the major allele, what's the minor allele, how many are missing, how many are heterozygous, things like that. And then this right here is now being called like this, and so this is using that Magritte pipe, and so this should bring the same thing. So again, we're taking that genotype, we're calling it site summary, and then we're getting the head of that. So we get the same thing. In addition to site summary, we can have a taxa summary table, which in this case, this is looking at sites, this is looking at taxa, so it's the same kind of principles apply. Is what's the sample ID name, how many sites are represented in each one of those samples, how many missing gametes are there, what's that proportion, how many heterozygous sites are there found for each one of these samples, what's that proportion, things like that, and also some inbreeding coefficients that are not presented right now in this table. Then we can also pull out, like let's say we want to get a vector of each one of our different taxa that are found within the the rTASSEL object. So we can take that rTASSEL phenotype genotype data set, and then we can use this taxa list, which is going to bring out the sample IDs. So in this case, it's 3316, 3811. Again, these are each one of those sample observations that are found within our data set, and that's just returned as a vector. We can also do the same thing for the position list. And this, instead of presenting a vector, it's going to provide some more information. So this is going to be kind of like more of a simplified version of the site summary. Namely, what's the name of the marker, where is it located, and what's the variant information. And I made a note during the workshop because, again, there was a lot of confusion about what is taxa. So taxa is synonymous with sample ID. And I'm just going to keep trying to reiterate that. And you can also think of it as like a genotype, it's like a clone, it's a variety. It's basically whatever you're calling a sample that's getting sequenced and getting variant information. So now we get into this next section right here, which is the analyses section. So besides getting summary information and loading data, we can perform various types of analyses, of course, with TASSEL. One thing that we can do is take a look at population structure. So we can, instead of looking at 3,000 or so variant markers, we can do dimensionality reduction and try to see, where is the variability being captured through principal component analysis, or PCA for short. And this is an rTASSEL method. And so when I call this rTASSEL, and then I call this double colon component, this is just basically from this library called this function. And if we take a look at the different parameters that you can specify within this, by default you can just pass in the object and that will provide you some default parameters. So if I call this PCA res and I take a look at the PCA res right here, it returns a list, and it's basically returning a list of data frames, right? So we've ran PCA. We have this PC datum, which will have each one of our sample IDs, And then by default, it will take a look at the first 5. It will return the first 5 principal components in your PCA analyses. And then you also have the eigenvalues and eigenvector components that are found here. These are data frame objects as well. So this would be like, for eigenvalues is for each PC, how much of the variation is captured. And then we also have the eigenvector components. And again, these two things are optional. You can return just like the PC datum or just that itself, or you can return the PC datum and the eigenvalues, or the PC datum and the eigenvectors, using all of these different arguments right here. And if you take a look at the rTASSEL website, this will provide you with more information about what each one of these parameters indicate. Because there's a lot of different arguments, and some of these, we have defaults for each one of these arguments, but in certain cases, that might just be fine. But if you want something more specific, you can then specify in the arguments, what you want. So maybe you want like the first 20 PCs, you can set that with like the number of components, for example. This report eigenvalues and eigenvectors, that would be either a true or false component, like do you want those reported or not, and other things. But for now, we're just going to take a look at the default settings. And for a lot of things, that's usually just fine. So we can then take a look at PC datum, take a look at that. So again, this is a data frame. And this data frame has a sample ID and then your first 5 principal components by default. And then what we can do is we can take that and we can plot it out. And so right now, at least for this workshop right here, I kind of showed a more customized way of plotting out PCs and then adding additional information to those PCs. Or at least in this, we are plotting a scree plot. And then we're also gonna plot like the actual PCA data down here, step by step, in more like kind of like a customized way. But later on in a newer release of rTASSEL, we're gonna provide helper functions that basically will do this automatically for you, making kind of like a more default generalized method of producing these PCAs and scree plots, that in case you don't want to go through all of the effort of taking this data and piping it into a ggplot workflow, this will automatically generate it for you. But keep that in mind, this will be default, it won't be customized. So if you want this for like, I don't know, a publication or for a presentation, this will be maybe something that you would want to also take a look at doing a more customizable approach. Because learning ggplot and learning how to visualize things, almost from scratch or from like a base component, is going to be very helpful for you, because you can tell a lot of different stories depending on the different types of data that you have. And in short, what we're doing right here is we're going to look at actually let's just calculate this first and then draw the figure. So this is a scree plot. And what this is showing is the first 10 principal components and how much variance is being captured in each one of those different principal components, right? And so we call that a scree plot. So generally, the first few principal components will hopefully collect a lot of the variability. So that implies that we have fairly decent dimensionality reduction. And then after those first handful of PCs, it kind of like almost flatlines. So what exactly are we plotting here? So we're taking from that PCA res, we're taking this eigenvalues datum. So I'll make another cell block above here to show what that is actually in terms of. So this is just, again, another data frame. I'll take the first 6 rows of that. So we've got, each one of these rows indicates a principal component that's being reported. And then you have the eigenvalue, and then you have the proportion of total variance captured. So for this, in this case, how much variance is being captured. And then as you go down here, what's your cumulative portion? Like once you start stacking up all of these different PCs, what is like the total combined proportion at, once you've stacked together 6 principal components. So we're just taking that data frame, we're piping it into this ggplot pipeline. So we're taking that PCA res, that eigenvalues data, and we're using, again, this magrittr pipe. So we're getting the data frame from the list object using the $sign operator. Then what we're doing is, since we're really only interested in this case for the first 10 PCs, and again, this is user defined, you can specify the first 20 PCs or the first 30 PCs if you really wanted to. It's up to the user. What we're going to do is we're going to take those rows. So we're just going to pass this head function, and the head has this n parameter that we're using for the number of PCs. So this would be 10. So we're taking the first 10 rows of that head, and then what we're doing is we're using another magrittr pipe. Now we're passing this into ggplot. And so this is where your actual visualization component starts. So this is from ggplot2. So everything that I'm highlighting right here is from ggplot2. We take that ggplot, so that's going to basically start the ggplot object. And then from that ggplot object, we populate it with different, geometries, different visualizations, different, different aesthetics that we can then visualize with our eyes. The next thing that gets passed is this AES function, which is an aesthetic function. And so one thing to note is that when you're piping things normally, you use this magrittr pipe. In ggplot, so again, all of this is ggplot2, instead of a magrittr pipe, we have to use a addition operator. And this is kind of like, there's a long story of why this is, and I don't really want to go into it in this video because this video is already going to be long enough. But long story short is that when dealing with ggplot, we use addition. When dealing with like everything else, we use this magrittr pipe. Okay. I mean, they're the same kind of like things, but it's just kind of like different time stamps in our history of when these things were developed. So we use this AES, we use the addition operator from that ggplot. We use this AES function, which has this x variable and a y variable. So those would be like your axes. So what are your axes that we're going to plot? So the x-axis is going to be the PC. So this would be PC1, PC2, PC3. You have to take note is that the current setup of rTASSEL, this is straight from the TASSEL 5 index, which is, it starts at 0 because all of the indices in TASSEL start at 0. So this is just being reported as is. But in a new update, this is going to be modified to be like 1, 2, and 3. Something that is more logical to a lot of users. So we're using that as our x-axis. So this is your PC ID, and then your y is what the y-axis is going to be. So it's going to be the proportion of total variance. So this would be this column right here. And then what we got to do is we have this group equals 1, because essentially this is going to be a line plot, and we have to specify that we set group as 1 because there is really no grouping whatsoever. This is just kind of like you can think of this as like a basic vector of data, and there's like no categorical information. So by setting that to 1 is like there's no categorical information. If there was, like let's say we had one line that indicates like, treatment A and another line that's like treatment B, we could indicate that group with like a treatment column. But if there is no like categorical information, we just set group to 1, and this is like, don't worry about grouping. The next thing that we pass that into is, so that's like just setting up the axes, right? The next thing that we do is we're going to pass that in using a geom line. So this would be like your geometry. So what do you want to actually visualize? So we want to make like a line chart here, right? Because we want to connect basically each one of these PCs together to see like the overall trend. So generally like line plots are more for like time series analysis, but you can also think of line plots as, like what are trends in the data, and like how do you want to generally like follow this? Like what is, if there is like a trajectory or momentum in like the visualization of your data. So in this case, generally scree plots will have like a line component because it allows you to visualize more of the trend over not time in this case, but in terms of number of PCs. So you kind of like, your eyes start from left to right, and then it looks like, oh, it's going down, So that's going to be one geometry that we add. And so we're going to set that geometry with a parameter within that function called color. And so that's just going to be like, what's the color of that line? In this case, we're just going to set it to red. And just remember, geom is short for geometries. And this is kind of like, the shapes. What are the things that are being distributed on the plot? The next thing that we have is geom point. So in addition to the lines, we're just going to add some points as well. You don't really need this. This is up to the user, and this is like the really powerful component of ggplot2. It's very, very customizable. Like if you don't want points, you can just disregard the points altogether, and now you just have like a line. But sometimes people like to have points because it helps indicate, okay, at PC2 we've dropped down to this, at PC3 we've dropped down to there. It kind of helps as like a more of like a visual statement of where each one of these data points lies within the line. And then we can set like a size parameter there. So we're setting that to size 3, and that's just basically the size of the dot. So if we do something silly like size equals 10, we're going to get giant dots, right? Or if we do like something like 0.5, we're going to get like microscopic dots. So again, that's up to the user and how you want to report that. But I think I just did by default size equals 3, because to me that looks okay. Then the last two things are just kind of like cleaning up the plot altogether. So we have this xlab and ylab. This is, lab is short for label. So I'll just put like lab equals labels. And then xlab, this is PC, and then ylab is proportion of total variance. Because like if you didn't have that, it would just call whatever the column name is in the data frame. So I guess, this column would actually be fine. But like if we, for example, just did this real quick, oops, wrong key bindings. I'll get rid of that plus for now. So you notice that instead of proportion of total variance, it's now proportion of total. And I'll return the addition operator. And now you notice that it's changed to proportion of total variance. So it's just making things a little bit more legible, a little bit more professional for a presentation or for a publication or report. And again, this is all like a notebook, and you can like compile this like HTML or whatever, or share with other users or your collaborators. Right, and so the next thing that we can do is, so this is a scree plot, but we can also plot like the actual PCs, and depending on what you're doing is, do you want to see structure or do you not want to see structure in your data, right? How, like, is there clustering? Is there, little communities forming in terms of like the variability in respect to the marker variant data? So instead of just like looking at dry tables of numbers, we can visualize that. We can visualize that clustering. And what I'm doing right here, and again, this is like the powerful thing of ggplot, is that we can start to add additional pieces of information to our data set. And so what I'm reading in is another example little data set, and this is called taxa metadata. And this is just some additional information for each one of these samples that are found within our data set. In this case, I am mapping, or in this case, I am associating with each one of these observations a categorical piece of information, which is subpopulation of maize varieties. So things like popcorn, mixed, non-stiff stock, tropical, things like that are found predominantly in different varieties of maize. So like, do each one of these subpopulations, how well do they group together, right? And you know, theoretically, they should group fairly well. And we can take a look at that by adding that to our PC data. So the first thing we're going to do is we're going to take that PCA datum. I'll just do the first 6 rows again, just to reiterate what that is. So we have the first the first 5 columns and the first 6 rows of the PC data. So these are your principal components. So this is through, you know, decomposition of multidimensional data. You get these PCs, and then what we can do is, if you take a look at these are essentially, you know, vectors of numbers, and we can do something like a scatter plot on a couple of these principal components. And so this would be like a, you know, your standard PCA plot. But in addition to that, what we're going to do is, you know, we're gonna add that metadata, or that subpopulation data, to help provide a little bit more context within what each one of these samples are and where they fall within the PCA plot. So we use this dplyr command, which is a join statement. So similar to how you join phenotype and genotype data in an rTASSEL data set, the same thing applies within things like dplyr, where you can join or find commonality between 2 different data frames. So in this case, we want to join this metadata, this subpopulation information, with this PCA data using this this sample column, or in this case, we're calling it a taxa column. So we can do different types of join statements. And so what we're doing is we're just doing a left join. And so what that is we're joining one thing by another thing. So we're joining the PCA data frame with this. So this will be like the first thing, and then we're joining it by this with this. And so we're taking this PCA datum, magritter pipe, left join this, that's the left hand side, with this, that's the right hand side. By what column? We're going to specify this taxa or sample ID column. So that'll be the first thing you do. All right, and so we can actually just run that to give you an example of what that looks like. And so what we've done is we just combined those. We merged them together. So we've combined them by this taxa column. So we just added that. And so you could just do something like a cbind, but that's a very dangerous operation, because, you know, what if the orders of taxa were different in one data set versus the other? If you just like naively bind row by row in each one of these samples, you could get like an observational mix-up, or like a sample mix-up, which would lead to incorrect results in analyses. So it's always good to do things like joins or merges in base R when you can. So the next thing we're going to do is we're going to pass that into a ggplot component. So the first thing we do is we pass that into ggplot, and we use this addition operator for each subsequent ggplot function. So the aesthetics in this case is going to be the X and Y axes are going to be PC1 and then PC2. But now we can also add this color component. So how do you want to color each one of these dots, right? So we're going to color it by subpopulation or category within this data set. Then we're going to pass just one geometry, which is geom point. and we're going to make each one of those points the size of 2. Okay, so we're going to run that, and now we get a visualization. So as you can see, we do have some structure, but another interesting thing to note is that, we get this kind of like triangle or like, upside down V looking object. But now that we've added that subpopulation information, we can now kind of like start to see that, oh yeah, all of the stiff stocks in this population for the most part tend to group together. All of the, I think that TS is tropical, group together, and then you have like this non-stiff stock kind of grouped together, and then you have some samples that just, we don't have metadata information, so those are just NA for now. So you do see clustering based off of, subpopulation, and again, each one of your analyses may be different. This is just, an example with some maze test data. And so what I'm trying to say is that, like, there isn't like one defined step of, or series of analyses that you can do to get a definitive answer, unfortunately. It's really up to, the question that you have in terms of, like, from biological support or, biological inquisition that you can use to then support whatever your visualizations or whatever your analyses are. So that's going to be up to the end user for that. Another thing you can do, besides PCAs, you can do things like phylogeny. And so instead of like collecting things by principal component decomposition, you can create phylogenetic trees based off of the genotype matrix. And so to do that, it's fairly simple. Is we use this create tree. And this has basically 2 parameters. It has your tassel object, which in this case is the genopheno object from before, and then what is the clustering method. And so the clustering method can be either UPGMA or neighbor joining, either one of those 2. And so these are 2 different algorithms. And so by default, it defaults to UPGMA. If you want to change it to neighbor joining, you can do that, but I'm just going to stick with this. We're going to pass it into this phylores object, which is going to be our results. So this would be our phylogeny results. So if I click on that, what gets created is this phylogenetic tree object with some additional information. So this would be 265 tips. So again, we're making a tree, so we have 265 tips. That's indicative of each one of the different samples that's found within the data set. And then we have different like combinations of nodes. So these would be like the combining points within the actual tree. Then we have the tip labels, which are just the IDs that we give each one of our samples. And then just some like some notes like this is unrooted, it contains branch links, things like that. But that's just by default because we're not really worried about that at the moment. So that's good and all, but like the nice thing about phylogeny is that we can actually visualize these trees. And so there's several methods that you can use to generate these visualizations. One of these is by using this create tree. So first, this create tree method is from rTASSEL, and this little plot or this little analyses right here is showing how we can take an object, pipe it into. In this case, we have this filter genotype table by taxa. So we have a series of different filtration methods that we can use in R. So we can like filter our data set by, minor and major allele frequencies. We can specify specific ranges within, like we just want these markers or we want these taxa in these cases. So this is just one example that I showed during the workshop, that you can then filter this down. Like let's say you're only interested in a specific set of taxa for whatever reason. This is just kind of like one working example that we came up with during the Senegal workshop. So yeah, we're just taking it to this create tree, which is making this phylogenetic tree object. And then what you can do is you can use base r plot method. So we run that and you get a mess. So if you take a look at this, you get, you get a tree, but it's very, very crowded because we have a little bit over 250 samples, right? And it's all being basically smushed or compressed into this little view space. And so if we go back to the very beginning of the workshop where I talked about this helper function, this is where this little helper function finally comes into place. So we've already set that up. And so what does this function allow us to do? If we go back down here, what we can do is we can set up, we can call that fig function, and then we can specify the width and the height of our visualization. So we can turn something like this that has a lot of data, and then we can kind of like stretch that out. We can make this image a lot taller or a lot longer in the notebook if we really wanted to. So we call that, and now we've taken this and we've stretched it out to something like that. Another thing that we can do is, so this is just kind of like using default plotting methods, and depending on the number of samples and depending on your approach, that might be fine. But we also have access to different libraries such as ggtree, which is, you can think of it as like ggplot for phylogenetic trees. And so you can actually take that object that's created by rTASSEL and then pass it into this ggtree library to help customize your plot. And what's really nice about that is, it gives you much more extensive setup in terms of the layout, in terms of like the shapes, in terms of like adding additional information, which is what we're going to do right here, and then coloring things, and just adding a lot more ggplot type control over something that is more basic in nature. So what we're doing is, we're just taking that object, making a tree, and then taking that tree object and passing it into this ggtree function. And this ggtree function will have, some just general kind of guides and layouts to how the tree is structured in terms of like the size of the lines, the layout. In this case, there's different categories that you can pick from. And, what I'm showing right here is not the definitive result of what you can do with ggtree. There's a lot more options, and I suggest you look up the documentation and read that if you're really interested in doing things with phylogeny in rTASSEL and the JupyterHub, because this is a very powerful visualization method, and you can do a lot of really cool, interesting things with it. So in this case, I'm going to change like this layout right here that's very long and obnoxious to something that's a little bit more compact through the form of a circle. And we're going to specify this none option for branch length. We're just going to kind of like, not make the branch length variable, but we're just going to kind of have, a more of a circular component. And then what we're going to do is, so from before, just to reiterate, we have that taxa metadata. We call that. We have this subpopulation information. What we're going to do is we're going to combine that with our ggtree data. And so again, you have this obnoxious additional set of user-defined operators here. So we have the Magritte pipe, which is surrounded by 2% and it's a right angle bracket. Then you have this this ggtree combine operator, which is unfortunately kind of annoying, but it is a left angle bracket with a plus. And again, this is user defined, so it has to be surrounded by these percentage signs. So this can, this looks kind of alien, but I mean, that's how ggtree does things. But with that being said, you can then add this additional metadata, and what that's going to do is it's already going to combine that with the metadata, or with the sample ID information that's found already in this genotype phenotype object, and it's going to automatically combine that together behind the scenes, where like you don't have to do like a left join, is what we showed before with plotting PCA. Is automatically going to do that. And as I talk about this could probably be something that, you know, we could also integrate in rTASSEL, now that I'm like talking to myself and thinking about this further. But yeah, so you can just like combine that with the ggtree object. It's going to do a union, or it's going to do a join behind the scenes, and then you can then pass these aesthetic components similar to ggplot, which in this case you can set axes, but in this case you can set like color, you can set shape, you can set all of these different things. And so one of these things is going to be color, and what are we going to specify? It's going to be the subpopulation type. So this is going to be the category that we're going to color by, similar to the PCA plot. So if we run that, and then also take note is that we're redefining our dimensional space in terms of our viewing area to just 7 by 7. So we run that, and we get something that looks like this. So we're coloring, so we have the size of the lines. We can actually, I made those a little bit thicker to help define the colors more, but like, you know, we can make those skinnier if we wanted them to. We can make them obnoxious if we wanted them to. Oof, that looks bad, but let's go back to 1.5. There we go. There's a reason why I picked 1.5. It's like the happy medium. So it helps accentuate the colors in this case. So again, similar to, I'm just going to comment this out. Similar to this, we get the same kind of grouping structure where you have, you know, the tropical lines and the stiff stalks, and then you have the, you know, the mixed, which is kind of like all over the place, and then you have the non-stiff stalks, things like that, all kind of like grouping together. And again, this is going to be based off of your data, again, similar to like the PCA thing. But yeah, that unfortunately doesn't really tell us anything. So color, so we're just, you know, this is just defining the color of each one of those end tips, right, by the subpopulation in this case. The next thing that we can do is calculate sequence diversity. So this would be calculating a different set of diversity metrics for each one of your markers. So basically how, you know, how diverse that actual marker set is. And so we can use this seek diversity component, or this function called seek diversity. And what this is doing is it's calculating sequence diversity, and depending on the size of your data, this can be as small or like finer grain resolution, or as, you know, big of a window as you want it to be. And so what we mean by that is, so first I'll go through each one of the parameters. So you have, first thing first is always going to be the object, the TASSEL genotype phenotype data set. And then you're going to have these 3 additional parameters. You're going to have this sliding window analysis. That's either going to be true or false. So if that's false, it's just going to generate, you know, a metric, like a combined metric across the entire collection of markers. And, you know, if you want to do that, that's up to you. But generally what people will do is they will do something like a sliding window. And so we set that to true, and that basically activates these next 2 parameters, which is the window size and the step size. And so what we mean by this is I've kind of generated this little text-based cartoon here. And what this is showing is that, let's say we have a window size in this case 200, for this example, we have each one of our markers down here. What is, if you're not really interested in doing it base pair by, or not base pair by base pair, but like marker by marker based analyses, if you do something like a window, what that's going to do is it's going to take, essentially, you can think of it as like an average for a given window of markers. So in this case, I set window to 3. What that's going to do is it's going to specify 3 markers within the entire collection. So we're going to always start at the first marker in this case, and then move down. And so that's where the step comes into play. So we have a window, in this case 3. We're going to take the first 3 markers that are in the index, analyze those, take the average, report that number, and then we're going to step. And so that's just going to essentially just move down the chain here. So we're going to go down to like the next marker. So we're, instead of starting at marker 1, we're going to be starting at marker 2 and then going to marker 4. Analyze that, get a metric, move down. Analyze that metric, get it, report it, move down, and you just keep doing that until you reach the end. So that's where window size and step size comes into play. And so we can set those, and so you can set these to how big of a window or to how small of a window, and how big you want your step size to be. So basically, how much do you want to keep moving down after each iteration in the diversity analysis. So we run that, and I'm going to look at the structure. So I'm going to call str. I'm going to call that object, and we get again another list of data frames. So and this is just the same as being reported within the TASSEL 5 API. The thing that we're really interested in is this diversity component, which is the data frame of here. We're just going to show, just taking the first 6 rows. And so what that's going to do is it's going to indicate, what chromosome this was found. So again, each one of those is like a window size of 200. So this would be like 200 markers. And then, this would be that would be like 1 to 200. This one would be 25 to 225, I believe, and then keep iterating. So start site, yeah. So 0 in this case, that would be 26 to whatever, and then 51. And you just keep iterating through that until you go through all the marker sets, or the entire marker collection in your data set. So you get the chromosome, you get the start position, and then you get the end chromosome position in this case. This is basically like where the physical location of that window is. Then you have the start site, and then you have the end site. So this is the size of your window. So that'd be, this again, index to start at 0 in TASSEL. So that would be 1, that would be marker number 1, and then marker number to 200. And then you have a mid site, that's just the midpoint, the median between the middle point in that window. And then how many are found, so window size 200, 200. And then average site count is like how many markers were found for each one of those, if there's NAs or so. And then you have the last 4 columns here, which is pi per base pair, theta per base pair, and then Tajima D. Haplotypes, this isn't currently available, but again, this is just being reported straight from TASSEL 5. But the things that we're interested in are the Tajima's D. You have the pi estimation, which is nucleotide diversity, and then the theta value, which are like 3 common diversity metrics that are found in population genetics. And if you want more information about that, if you go to the rTASSEL website, actually I could just show that right now. yeah, if you just go to the actual website. So this is the actual rTASSEL website. This is found at maze-genetics-genetics.github.io/rTASSEL. And if you go to reference, and then if you search for sequence diversity, and actually what you can do is you can go here in the search panel, and you can actually search for it. You'll be provided with further information about what each one of these are. So pairwise divergence, segregating sites, estimated mutation rate, so theta, and the Tajima's D will be calculated. So again, if I'm not explaining something in thorough for this video, just remember the website will have additional information. And then, if something doesn't make sense, feel free to reach out to me and we can chat. So we can generate a table, but then we can also pass that into a figure. And just like the other examples, we're going to have some default, report generators for these figures in a future update. That's currently still being tested out. But for now, what we just showed for the workshop, at least at the time of the workshop, those weren't really initialized. We're just passing this through like a normal ggplot pipeline. So we're taking that table. What we're just doing for now, in this case, for this figure, is we're filtering the first chromosome, in this case chromosome 1, because it'd be kind of silly to compare chromosome 1 and 2 or whatever, or try to have everything all in one plot or just like one chart. So we're just filtering it down by the chromosome. So we're selecting chromosome 1, all of the different windows that were found there, piping that into ggplot, doing the aesthetic. So in this case, start chromosome position, and we're just doing Tajima's D. We're doing a line and a point again, and then we are doing by position in Tajima's D for the XLab. And so here you get basically a position informational reference across the entire chromosome, starting at position 0, going all the way up to 3 times 10 to the 8th. So basically spanning the entire chromosomal arm, and then you have like the position of the window of where those markers are, and then calculating the metric for that window. In this case, we're just reporting Tajima's D. So again, this is just indicating, based off of what Tajima's D is reporting, you can either have like higher positive values and higher negative values, indicating different diversity components. But yeah, so this is just like one way that you can plot, and then you can do chromosome 2 if you wanted to, or like whatever chromosome that you want. You can plot them all together using a facet component within ggplot, but that's for another video. And then the last component here that we wanted to talk about, at least for this first session, was we can also do things like GWAS, or simple association analyses. So again, just to reiterate, we have this TASSEL genotype-phenotype data set that has both genotype information and phenotype information. We can combine those two together to associate the trait with genotype and or different covariate components. And then what we can do is we can pass that through this rTASSEL method called rTASSEL, or association or assoc model fitter. And what this is going to take is it's going to take, of course, the data set, and then it's going to have this parameter called formula, which is going to be the trait by whatever else that you want to call it in terms of like covariate and or factor components within your data set. The next thing is like, from this you can, if you just like set this and you don't fit markers, You can generate basic linear unbiased estimators. So basically generating a fancy average for each one of those samples, because remember we had different location components within our experimental data, so you can like compress that down to like one value and then feed that into something else. And then we have this fit markers component, so that's if it's set to false, and then if this is set to true, it will fit the actual markers within the genotype data to this formula. So this will be essentially adding the G matrix to your linear model. And then we can also report the maximum P value that we want to report. So we can like set a hard threshold if we're like, worried about, overloading our environment or our session with, tons of data. So if like you're dealing with a marker data set that's like several million different markers, you probably don't want to be dealing with a data table or data frame that's several million rows long. You can do like a threshold and report, just the significant p-values in this case. I'm just setting this to 1 because I just want to report everything for this next component that I'm plotting here. So yeah, this is, and again, this formula component is looking at just like a simple, it's basically emulating the same like formula that you would do in things like, linear model in base R. So like, if we have the iris data set, right? This is just a built-in native data set to R, which is looking at different observations across different species of this iris genus. This is just a flower, so we're measuring different components within this flower. We can do something like a linear model formula, and that would be things like, we have the formula component. So let's say sepal length, I can spell sepal length is going to be dependent on petal length. The data in this case is going to be the iris data set. We can generate a linear model. So like it's basically emulating that kind of component where this component right here is your y, and that's your that's your x. So the very base sense, you have your y variable and then your x variables. All right, so you run that. It's going to first calculate some blues and then use those blues to feed into the GLM for fitting markers. And so we can take a look at what that is. And again, it's just a list of data frames. So these are table reports within rTASSEL. And so we got our actual stats and then some additional statistics on the genotypes. And then what we can do is we can take that stats data frame and then we can pass that into another rTASSEL function called Manhattan plot. And what that's doing is it's visualizing each one of the p-values in the fit, or essentially the negative log 10 of each one of those p-values. So if we run that, it's taking the p-value for each one of those 3,000 or so markers for each one of those independent fits of GLM fits. So it's extracting the p-value, and we're taking the negative log 10 of that p-value because, p-values are going to be a value between 0 and 1, and the more significant values are going to be compressed to 0. So it wouldn't make for a good plot if we were to do that, so we just take the negative log 10 of said thing. So like the more significant, I guess, a value is going to be. So if we take the negative log 10 of, let's say, 0.0001, you're going to get the value of 5 because we're taking base 10. We're going to have 5 components there. So it's just taking like incredibly smaller numbers and then inverting it and making them bigger numbers, so you can get these peaks. And the reason why it's called Manhattan, because preferably if you were to run this, you would like to see these significant peaks of different data points across the markers that kind of, you know, if you squint a little bit, it kind of looks like the skyline of Manhattan in terms of like each of all of the many different types of skyscrapers that are found in Manhattan. That's where the name comes in. So again, this is like a diagnostic plot. If you really wanted to, you could take that data frame and then pass it into you know, a ggplot workflow, as I've mentioned before, or you can just generate the plot as is. And so this is, you know, automatically generated for you. You can set the threshold, and this is like the negative log 10p threshold, and we can plot based off of given traits. So in this case, I'm just plotting ear height. You know, if I really cared, or, you know, if I wanted to like loosen that threshold, like instead of like saying negative log 10 of something like this, you know, you could do something like, I don't know, 2, and then you can replot that. And now that threshold line goes down further, and then the number of significant, in quote, points, data points, in this case, for further inspection would increase. So those values would increase in size in terms of its point, and the line goes down to 2. So by default, this is set to 5. And this is just taking a look at those stats if you wanted those. And yeah, and so, and then if you wanted to, you could, a typo, it should be significant peaks. So if you wanted like significant, you know, from that data set, and you wanted just to report each one of those markers that was significant, you could filter. So you could use something like dplyr and then filter based off of p-value, and that would give you your respective hits. So in this case, this toy example, we got 3 kind of significant hits in this case of our model. So 2 found on chromosome 1, and then 1 found on chromosome 10. But again, this is, you know, not like the end all be all. Further information, further validation, of course, would have to go into this. And then we can also, if you really wanted to, you can export the data. So you can take like the GLM stats and you can write it to like a CSV file using something like Reader or, you know, base R or whatever like writer method that you want to use. So, because I mean, this is essentially a data frame in R, and I think people watching this will already have like a good idea of what a data frame is, at least not from this, but, you know, maybe from your own learning or from like the past virtual workshop that we hosted before the Senegal workshop. Same thing, the same logic applies to that, you know, because this is, again, this is just a data frame, and we can, you know, write that out, and we can pass that into anything else. So that is the end of this video. In the next video, we're going to be going through day 2 of the Senegal workshop, where we are going to be talking about, if this loads, we're going to be talking about some additional information about rTASSEL. We're going to be going through some additional steps of that. And then we're going to be talking a little bit about QBMS moving forward in terms of like data management systems, and then how you can do genomic prediction using rTASSEL and Summer combined within a notebook to get predictive measurements. But that is going to be for video 2. So I will see you in part 2.